Two maps of the same country hang on the wall. The first is a satellite photo projected flat every mountain, river, and city in its measured position. The second is an artist's sketch. Rivers bend in impossible ways, oceans shrink, but every city sits next to its true neighbours. Same data, different projections, different stories. PCA and UMAP are these two maps. Understanding when to read each one is the most important skill in exploratory data analysis. The satellite photo. A camera in orbit captures the country from above. A cartographic projection like Mercator flattens the curved surface onto a flat page. Projection is linear. Straight lines on the globe become straight lines on the map. Distances between major cities are roughly proportional. Continent shapes are recognizable. But there's a price. Greenland appears the same size as Africa, even though Africa is 14 times larger. The linear projection preserves large-scale structure but distorts at the edges. Is this projection linear, distance preserving, globally accurate, locally distorted? What does PCA preserve? Global distances on the satellite map. The distance from Tokyo to London is proportional to the actual distance. In PCA, the distance between two samples reflects their overall difference across all features. Global structure. The continents are in the right places relative to each other. In PCA, major clusters, tumor versus normal, immune versus epithelial, are separated proportionally to their true biological differences. Variance. The biggest features dominate. On the map, the Pacific Ocean is the largest feature. In PCA, the principal components with the most variance capture the largest biological differences. What does PCA distort? Local neighbourhoods. A small island chain near the coast might get projected away from the mainland because the linear projection can't capture the Kirk coastline. In single-cell data, rare cell subtypes might overlap with unrelated cells on the PCA plot. The linear projection can't unfold the manifold. Nonlinear relationships. A mountain pass connecting two valleys through a winding path appears as a straight-line distance on a flat map much shorter than the actual route. In biology, a differentiation trajectory that curves through gene expression space gets compressed by PCA's linear projection. In single-cell biology, PCA is the first step. 20,000 genes measured in 50,000 cells. Pair reduces to 50 principal components. On the PC versus PC scatter plot, major cell types separate. T cells on the left, B cells in the middle, monocytes on the right. These are the continent's large-scale separations that PCA captures perfectly. But within the T cell cluster, CC positive and regulatory T cells overlap. These subtypes differ by subtle gene expression changes, small islands near the T cell continent, and because linear projection can't resolve them. The satellite photo shows continents clearly but blurs the neighbourhoods. When should you use the satellite photo? Quick overview. What are the major clusters in my data? Quality control. Do samples from different batches separate on PCA? That's a batch effect. Distance measurement. How different are treated versus controlled samples? The distances are interpretable. And critically, preprocessing for downstream methods. Run PCA first to reduce noise, then apply UMAP to the PCA output. The satellite data feeds the artist. PCA before UMAP is standard practice because UMAP on raw 20,000 dimensional data is slow and noisy. The satellite photo, global distance is preserved, linear projection, the big picture at the cost of local detail. It captures the continent's major biological differences, but can't unfold curved neighbourhoods. It's the foundation. Next, the artist's map that shows what PCA can't.